Okay, so welcome back to this next video in which we are discussing Cys-Loop receptors, or Cys-Loop ligand-gated ion channels if you prefer. Okay, so in this video what we're going to do is move on to discussing another family of Cys-Loop receptors that are present within humans and other mammals, which are the glycine receptors. Okay, so again, this is a family of Cys-Loop ligand-gated ion channels. Okay, so all of its members are going to have that same structure that we've discussed now. Okay, and they're going to be involved in inhibitory neurotransmission within the spinal cord. So we've discussed how GABA is one of the major inhibitory neurotransmitters within the brain and also the spinal cord. Okay, so when you release GABA onto a postsynaptic neuron, it will open GABA A receptors, and this will allow in an inhibitory chloride current which will hyperpolarize the cell and make the postsynaptic cell less likely to fire an action potential. Okay, now in the spinal cord you also use glycine as an inhibitory neurotransmitter, and again it's going to act on ligand-gated ion channels, which are Cys-Loop ligand-gated ion channels, to open a chloride channel which will allow in an inhibitory chloride current into the cell and therefore hyperpolarize the postsynaptic cell, making it less likely to fire an action potential. Okay, right. So, just like we've discussed for the nicotinic acetylcholine receptors and the GABA A receptors, there is not just one glycine receptor. Okay, again, it's an entire family of receptors. Okay, and the reason that you have more than one glycine receptor is that there isn't just one subunit to make up glycine receptors. Okay, in humans we actually have 5 different subunits to make glycine receptors, and that means that we can build different pentamers out of these. Okay, so let me start by telling you about the different subunits for glycine receptors. Okay, so again, the naming is very similar to what we've seen for the subunits of GABA A receptors and nicotinic acetylcholine receptors. Okay, so you have 4 alpha subunits that are called alpha 1, alpha 2, alpha 3, and alpha 4. Okay, so those are the alpha subunits of the glycine receptors. And then the final subunit, because remember I told you that there's only 5 different types of glycine receptor subunit, is a beta subunit, and that's just called beta 1. Okay, so slightly less subunits for glycine receptors than there are for nicotinic acetylcholine receptors or GABA A receptors. Okay, right, so those are the subunits out of which we can build these glycine receptor pentamers then. The question now remains, how do we actually assemble these subunits together in a pentamer? Okay, what sort of arrangements can we produce? Well, glycine receptors have not received the same level of study as GABA A receptors and nicotinic acetylcholine receptors, but what we do know is that out of all 4 of the alpha subunits you can build Homo Pentamers. Okay, so all 4 of these alpha subunits can be used to create Homo Pentamers, and I'll just draw a basic picture of this in a moment. Okay, so what you'll do then is you'll take 5 identical alpha subunits. Okay, so you'll either pick alpha 1 or alpha 2 or alpha 3 or alpha 4. You'll pick one of those types. You'll make 5 identical subunits. Okay, and you'll stick them together to make your Homo Pentamer, and this is what it will look like from above. Okay, so all of these are an identical alpha subunit. So let's say I picked alpha 1. Okay, so I'd stick alpha 1 now in all 5 of these sockets like so to create me an alpha 1 Homo Pentamer. Okay, and you can do this for absolutely all of those 4 alpha subunits. It's not just alpha 1 that you can do this for. All of them can form Homo Pentamers like this. Okay, so where will the ligand binding sites be for these Homo Pentamers? Well, we know that the ligand binding sites in Cys-Loop ligand-gated ion channels are always at the interface between neighbouring subunits. Okay, and by the beautiful symmetry of these Homo Pentamers, we therefore know that we must have 5 ligand binding sites for glycine, 1 at each of these interfaces between neighbouring alpha 1 subunits like so. So each one of these purple dots must be a binding site for glycine, basically. Okay, so we can form these alpha subunit Homo Pentamers which bind 5 glycine molecules, and of course when those glycine molecules open, the channel will open with the gating mechanism that we've seen, okay, and then it will allow chloride anions to move into the cell, so it's a chloride selective ion channel, and therefore will have rings of positive charge along the pore. Okay, right. Now the alpha Homo Pentamers aren't the only type of glycine receptor that we know that you can build. We also know that you can build Hetero Pentamers, okay, where you pick one type of alpha subunit and then you combine it with the beta subunit, basically. Okay, so you've only got one choice of beta subunit, that's the beta 1, but you can choose any one of the alpha subunits you like to pair the beta subunit with. So let me now show you how you can build one of these alpha-beta Hetero Pentamers. Okay, so basically what you're going to do is you're going to take 2 of your chosen alpha subunits. Again, let's imagine that I choose alpha 1. So you'll make 2 copies of the alpha 1 protein, okay, and then you're going to combine those 2 copies of the alpha 1 protein with 3 beta 1 proteins in the arrangement that I'm about to show you here. Okay, so again, I'll draw this from above here. So we're looking down from the extracellular side, okay. Here are the 5 separate subunits making up our glycine receptor, okay, and I've said you're going to use 2 alpha subunits that are identical and then 3 beta subunits. So the sort of common sense thing to imagine that it will be the case is that the alpha subunits will be separated by a single beta rather than having them next to each other. Okay, so that is indeed how we think this arrangement works. You put your 2 alpha subunits separated by a single beta here. Okay, so I'll colour this in. So we'll colour in the beta subunits here in red, okay, and then we'll colour in the alpha subunits in a moment in green. Okay, and I've chosen alpha 1 there, but it's believed that you can do this with any alpha subunit, so you can pick alpha 1, alpha 2, alpha 3, alpha 4, whichever one you like, basically. It's just important that both of those alpha subunits are the same type, basically. Okay, now, the binding sites then for these alpha-beta Hetero Pentamers for glycine are still controversial. We don't know, basically, how many glycines can bind, whether a glycine can bind at every single interface, or whether there are certain interfaces which actually work and bind glycine and others which don't. That's still controversial, basically. So we don't know how many glycines at present bind to these alpha-beta Hetero Pentamers. Okay, but the answer is, of course, some glycines do bind there, and they will trigger the opening of the alpha-beta Hetero Pentamer here. Okay, right, so there are our glycine receptors. They are pentamers of these glycine receptor subunits, okay? They open in response to glycine, and they're going to allow chloride anions into the cell when they open, and therefore they're going to cause an inhibitory current and make it less likely that the postsynaptic cell will fire an action potential. Okay, right, so they're used within the spinal cord. What I now want to talk about is a key example of where they are used in the spinal cord, okay, and that will motivate hyperekplexia and the symptoms of hyperekplexia, which is where you have problems, mutations, germline mutations in subunits for glycine receptors, okay, and by looking at this example of where glycine receptors and glycinergic neurotransmission is actually used, we'll be able to motivate the symptoms of hyperekplexia. Okay, so basically, in the spinal cord, you have the cell bodies of motor neurons, basically. So let me draw this here. So let's say this is a cell body of a motor neuron here, and I'll just draw the spinal cord here. So here is the spinal cord. This is the ventral aspect, and this is the dorsal aspect. Okay, and here is our alpha motor neuron, which will send its axon out of the spinal cord and will eventually go off and innervate some skeletal muscle cell here. And we've studied this neuromuscular junction now because that's where the nicotinic acetylcholine receptors were one of the very notable examples of a nicotinic acetylcholine receptor is involved. Okay, right, so this is representing our alpha motor neuron here with its cell body in the spinal cord and its axon terminal synapsing onto some skeletal muscle cell here. Okay, so neuromuscular junction there. Okay, right. So this is an alpha motor neuron. I'll just label it up. Alright, so basically we've discussed that skeletal muscle cells receive a certain amount of tonic stimulation to contract, basically. Alpha motor neurons are firing at a tiny little rate usually, okay, and they do cause a little bit of tonic contraction in skeletal muscle tissue, basically, okay, and this is what's known as muscle tone, okay. Now, basically there are neurons in the spinal cord that are inhibiting the alpha motor neurons and making sure that they don't fire too often, basically, making sure that the muscle tone is kept down, basically, and that really the alpha motor neuron is only going to fire an action potential when it receives command, when it receives a lot of excitation from higher centers, okay? So basically these alpha motor neurons have inhibitory neurons, which also have their cell bodies in the spinal cord, which are all the time tonically inhibiting these alpha motor neurons and trying to make sure that they don't fire action potentials at a too high rate, basically. Okay, and these neurons here, these are known as inhibitory interneurons of the spinal cord. Okay, so in turquoise, this is an inhibitory interneuron. Okay, so these inhibitory interneurons have these synapses onto the alpha motor neurons, which are inhibitory synapses. Okay, and the neurotransmitter used in these inhibitory synapses is glycine usually. Okay, so they are releasing glycine onto the alpha motor neuron, and therefore the alpha motor neuron has glycine receptors on its surface, which we've just studied. Okay, and we know how this will work now. The glycine receptors will allow chloride currents into the cell, which will inhibit the alpha motor neuron from firing an action potential. Okay, so the alpha motor neuron has a lot of these inhibitory interneurons synapsing onto it, which are making sure that it doesn't fire at a tonic level too high, basically, making sure that muscle tone is kept at a very low level, basically. Okay, and therefore that this alpha motor neuron is only really going to start firing action potentials at any sort of a decent rate once it receives command from higher centers. Okay, so from upper motor neurons, pyramidal neurons descending in the corticospinal tract. Okay, right. That's an example of where glycine receptors are really important in keeping muscle tone down by stopping alpha motor neurons from firing too much without command from higher centers, basically. Okay, right. So now what I want to talk about is this clinical correlate of glycine receptors, which is hyperekplexia. Okay, now, in hyperreplexia, you have germline mutations in your subunits for glycine receptors. Okay, so in some of these subunits for glycine receptors, you're going to have mutations, basically, in them, in the germline. Okay, and that's going to trigger loss of function of glycine receptors. So they're going to be loss of function mutations in these glycine receptor subunits. Okay, and when you get loss of function in your glycine receptors, then that is going to mean that these synapses become less effective, basically. Okay, how good the inhibitory interneuron is at inhibiting the alpha motor neuron is going to be weakened, basically, because now it's going to be releasing the same amount of glycine onto this alpha motor neuron, but now the alpha motor neuron is less sensitive to that glycine. It's going to let in less chloride current for that same amount of glycine, and therefore that tonic inhibition that the alpha motor neuron is receiving is going to be tamed, basically. It's going to be reduced. Okay, so now it's going to be easier for that alpha motor neuron to fire action potentials, basically, and therefore what ends up happening in hyperekplexia is you end up with far too high muscle tone, okay, which is called hypertonia. Okay, and hyperekplexia is also sometimes called stiff man syndrome. okay, because the hypertonia, too much muscle tone going to all of your skeletal muscles, okay, so it's not just to a specific skeletal muscle cell, it's to all of them, okay, agonist muscles and antagonist muscles. So for instance, biceps and triceps are now going to be contracting at a higher level tonically, okay, and what it ends up producing is it makes your muscles very, very stiff basically, because they're contracting a little bit all the time at too high a level basically. So if you try and move the joints of someone with hyperexplexia passively, okay, so like so, you'll just sort of move their joints, it'll be really, really difficult. Their joints will be really stiff because their muscle cells have got this too high tension in them basically, they've got too high muscle tone. Okay, so hypereplexia then is also sometimes called stiff man syndrome. Okay, and it results from these germline mutations in subunits for glycine receptors basically. Okay, right, so that's all I've got to say on glycine receptors. Let's now move on to the final family of cis-loop ligangated ion channels present in humans, which is the 5-HT3 receptors, okay, or the type 3 serotonin receptors. Okay, so 5-HT3Rs. Right, okay, so again, this is not just a single type of receptor, this is a family of receptors, okay, and they all respond to their ligand, which is serotonin, which we discussed right at the start here. Okay, so here is their ligand, which is 5-HT or serotonin. Okay, and we discussed at the time that these receptors are all going to be permeable to sodium and potassium. Okay, so cations, and therefore the main ion that's going to move when you open one of these channels in a cell membrane of a normal sort of resting membrane potential is going to be sodium, which is going to move into the cell, okay, down its electrochemical gradient. And therefore you're going to produce depolarization of the postsynaptic cell. Okay, right, so again, the first thing we want to discuss is the fact that this is not just a single receptor, this is a family of receptors. Okay, so we want to look at how many different subunits you have for 5-HT3 receptors, and then we want to discuss what is known about how these subunits for 5-HT3 receptors can be combined together to make pentamers. Okay, right, so there are 5 subunits for 5-HT3 receptors that are known, just like there are 5 subunits for the glycine receptors that are known. Okay, and these are called the 5-HT3A, the 5-HT3B, the 5-HT3C, the 5-HT3D, and the 5-HT3E subunit. Okay, so 5-HT3A is one subunit, the 5-HT3B is another, 5-HT3C, etc. And that gives us a total of 5 different subunits that we can use to build 5-HT3 receptors. Okay, now, these last 3, 5-HT3C, 5-HT3D, and 5-HT3E, very little is known about them. We know they exist, okay, but as far as I know, so far we do not know whether or not they can actually form pentamers. Okay, I don't think it has yet been found that they do form pentamers, okay, and in fact we only really know about 2 different types of 5-HT3 receptor pentamers, okay, which I'm now going to tell you about. So the main one that we know about is the 5-HT3A Homo Pentamers. So Homo Pentamers are always really simple, okay, so again this is just 5 identical 5-HT3A subunits joined together to make a 5-HT3 pentamer here. Okay, so I'll draw it just to make it crystal clear. Okay, so these are all now 5-HT3A subunits. Okay, and I won't put the 5-HT bit because it won't fit in. Whoops, what have I done? I can turn that into an A. Okay, 3A here and 3A here. So this is what we would call a 5-HT3A Homo Pentamer, and it's known that these things exist and that they're important. Okay, now again, as always with Homo Pentamers, it must contain 5 ligand binding sites for 5-HT, basically, 1 at each of these interfaces. So each of these green dots represents a place where 5-HT or serotonin can bind to the 5-HT3A Homo Pentamer, basically. So the 5-HT3A Homo Pentamer has 5 ligand binding sites for 5-HT. Okay, and again, when that 5-HT binds, it will trigger the channel to open by the mechanism that we've looked at. Okay, remember the M2 helices twist, okay, breaking the hydrophobic girdle and therefore causing the widening of the channel. Okay, and it's gated by 5 serotonin molecules, and when it opens it will allow in sodium ions into the cell and therefore activate the postsynaptic neuron to fire an action potential. Now, the 5-HT3A Homo Pentamer is not the only type of 5-HT3 pentamer that we know exists. We also know that a Hetero Pentamer exists, but very little is known about this Hetero Pentamer. Okay, so there's a Hetero Pentamer called the 5-HT3AB Hetero Pentamer. Okay, and basically in this Hetero Pentamer you have 5-HT3A and 5-HT3B subunits, but we don't actually know the stoichiometry really, okay, and we definitely don't know the arrangement of the 5-HT3A and 5-HT3B subunits, and even less do we know how many binding sites it has. Okay, so at present all we know is that what you can form is pentamers which contain 5-HT3A and 5-HT3B subunits. That's all we know. We don't know what the stoichiometry is, or, well, there are obviously suggestions of what the stoichiometry is, but everyone has their different suggestions, so it's still controversial as to what the stoichiometry is. Okay, we definitely don't know what the arrangement of the 5-HT3A and 5-HT3B subunits is in the pentamer. Okay, and then ligand binding sites, of course, that's still controversial as well. So at present we know that a heteropentamer forms, which involves 5-HT3A and 5-HT3B subunits, but we don't know much more about the intricacies of its structure, basically. Okay, right. So these channels then, they open in response to serotonin, and they're going to allow sodium and potassium ions to move through them, but the important one is sodium is going to move into the cell, and that's going to be an excitatory current. Okay, right. So now what I want to talk about is a clinical correlate of the 5-HT3 receptors, okay, which is the fact that 5-HT3 receptor antagonists exist, okay, and they are very clinically useful. They are used as antiemetics, basically. So I want to talk about 5-HT3 receptor antagonists. Okay, so basically these are drugs which can bind to the ligand binding sites of 5-HT3 receptors, so these binding sites here, and which will not stimulate the opening of the channel. So they bind to the same site as 5-HT, but they don't stimulate the opening of the channel. They just block the 5-HT from binding there, basically. Okay, so let me give you an example of a 5-HT3 receptor antagonist. The main example is ondansetron. Okay, all of the 5-HT3 receptor antagonists end with this suffix zetron. Okay, but ondansetron is one of the main examples. Oops, what have I got to put that S in there for? Get rid of that. Okay, ondansetron is one of the major examples. Okay, I've put the S there because the 5-HT3 receptor antagonists are also sometimes referred to as the zetrons. Okay, so be aware that if you hear people talking about the zetrons, that just means the 5-HT3 receptor antagonist, and a major example of the zetrons is ondansetron here. Okay, right. So basically, what are these drugs used to treat? Well, they're used as antiemetics. Okay, so they're used to treat vomiting, specifically vomiting that's induced by anti-cancer chemotherapy. Okay, so often when you take horrendously cytotoxic drugs as part of anti-cancer chemotherapy orally, then it can trigger vomiting, basically. Okay, and this is one of the side effects that people dread with taking anti-cancer chemotherapy, that it will induce them to vomit, basically. Okay, and 5-HT3 receptor antagonists such as ondansetron can be given with the anti-cancer chemotherapy, and they're incredibly good at stopping the actual vomiting that you get when you take anti-cancer chemotherapy. They're not as good at stopping the nausea that you get, okay, the feeling of wanting to be sick, but they are very good at actually stopping you being sick. Okay, so they do have some good qualities. Okay, so what I now want to look at is how do these drugs stop you vomiting in response to chemotherapy. Okay, and therefore we're going to see a physiological function of 5-HT3 receptors. Okay, right, so let's go over the page here. So, firstly, what we need to discuss is why are you sick when you take anti-cancer chemotherapy, okay? What's the actual pathway from you taking the drug and it going into your intestine to you actually vomiting, okay? So, basically, this involves, or at least this is believed to involve, serotonin big time, okay, and that would make sense since the 5-HT3 receptor antagonists are so effective at stopping the vomiting, okay. What's believed to happen is if I just draw the gastrointestinal system here, so let's have the esophagus here, okay, then we'll have the stomach here, then we'll have the beginning of the duodenum here, okay, like so, and I don't think we'll need to go much further than that, that'll do. Okay, so, basically, in the wall of the small intestine, okay, and this is the first portion of the small intestine known as the duodenum, okay, in the wall of the small intestine you have a special type of cell, okay, which I'll draw here, okay, which are responsible for checking that what you are consuming, what is in the lumen of the intestine, is not poisonous, basically, okay, and these are called enterochromaffin cells, okay, so this is an enterochromaffin cells. So the job of these enterochromaffin cells is to check that what you are eating is not poisonous, basically. Now the problem is that when you take anti-cancer chemotherapy, many of these drugs are incredibly poisonous. They're cytotoxins, okay? So the enterochromaffin cells do not like the anti-cancer chemotherapy. So let's have the anti-cancer chemotherapy represented as these dots here. Okay, and I'll colour those dots in purple. So this is our drug molecule coming here in the intestine. We've taken the drug molecule, and now the enterochromaffin cells are going to be exposed to this drug molecule. Okay, and they're not going to like it, so they are now going to initiate the signal that leads to you vomiting. Okay, and it's believed that one of the key things that they now start to release is serotonin, basically, 5-HT. Okay. Now, then there are two ways in which this serotonin is believed to actually cause vomiting. The first is that it can actually, it's believed to actually act on sensory neurons that are very nearby the enterochromaffin cells, okay? Specifically sensory neurons which will have their axons going up in the vagus nerves. Okay, so here is a sensory neuron's axon terminal, and it's believed that these sensory neuron axon terminals have 5-HT3 receptors on them, basically, on their surface. So this is believed to be a 5-HT3 receptor, although this is still controversial whether these axon terminals of the vagus nerve have 5-HT3 receptors on the surface. So I'll just colour that 5-HT3 receptor in. So here is the 5-HT3 receptor in turquoise here. Okay, right. So this is a sensory nerve here, which will have its axon going up the vagus nerve, or one of the vagus nerves, because remember you have two, one on either side, the left vagus nerve and the right vagus nerve. So this is the vagus nerve, or cranial nerve 10. Okay, so I'll just draw it like that. It's only one of the axons in the vagus nerve, of course. Okay, so this is cranial nerve, and what's the Roman numeral for 10? There we go. Okay, right. So basically this sensory neuron will have its axon terminal near the enterochromaffin cell, and it's believed that it has 5-HT3 receptors on its surface. And then, of course, the serotonin that's been now released by the enterochromaffin cells can activate these 5-HT3 receptors, trigger a sodium current to enter that sensory nerve's axon terminal, and then the sensory nerve will fire an action potential up the vagus nerve, and it will go up to the brain, and it's believed to activate centers in the brain, which are then going to actually drive vomiting. Okay, and we'll just abbreviate these down to vomiting centers. Okay, so the signal goes up to the brain, activates special centers in the brain which coordinate vomiting. Okay, which are in the brainstem and are called vomiting centers. Okay, so we'll just have those represented as a box. Right. Okay, so that's one way that 5-HT is believed to cause vomiting. It's also believed that the 5-HT is actually going to go into the blood and is going to act on an area in the brainstem that's very close by to the vomiting centers, and which activates the vomiting centers as well. Okay, and this special area which the 5-HT is believed to activate is what's known as the chemoreceptor trigger zone. Okay, now let me show you where the chemoreceptor trigger zone actually is. So basically it's in the medulla. Okay, so let me just remind you of the structure of the brainstem in case you're not familiar with it. Okay, so I'll draw this down here. Okay, so if we have the spinal cord here, okay, the spinal cord will pass through the base of the skull through a large hole called foramen magnum. Okay, so this circle here is representing foramen magnum, which is a big hole in the base of the skull. Okay, so the spinal cord is below that, and now when the spinal cord, coloured here in green, passes through the foramen magnum, it then becomes the brainstem. And one of the first structures of the brainstem is a portion known as the medulla. Okay, so this portion here, which is now inside the skull, this is called the medulla. Okay, so this is the first portion of the brainstem basically, and then above the medulla you'll have other structures like the pons, and then on top of the pons you'll have the midbrain. Okay, but it's only the medulla that we need to know. So the medulla is the continuation effectively of the spinal cord inside the skull basically. It's the first bit on top of the spinal cord. Okay, now basically what I'm now going to do is take a cross-section through the medulla and have a look at it. Okay, and then I can show you where the chemoreceptor trigger zone is. And we're taking a cross-section quite high up the medulla. Okay, so basically when you take a cross-section through the medulla, it looks like this. Okay, you have these projections backwards, which are connections with the cerebellum, which is a portion that's going to be behind the medulla here. Okay, then you have these portions at the front here which are going to contain the corticospinal tract. Okay, and then right at the back we have another one of those on the other side, like so. Okay, so let me just highlight up some important portions of the medulla. So this is the front of the medulla, this is the ventral aspect down here, and this is the dorsal aspect here. Okay, right, so these little processes at the front here, these little portions here which I'm now circling here, and which I'll colour in blue, they are what are known as the pyramids, and they contain all of the axons that are coming down from the cerebral cortex, okay, to innervate alpha motor neurons and tell them to fire action potentials. Okay, so basically these are bundles of axons which are sending messages which are the instructions of how to move basically. Okay, and they're known as the pyramids. Okay, or the pyramidal tracts basically. Okay, and basically they're just the portions of the corticospinal tract, which is this bundle of axons moving down from the cerebral cortex to alpha motor neurons in the spinal cord to tell them how to fire and therefore how the body should move. Okay, and then just that portion of the corticospinal tract which goes through the medulla before it enters the spinal cord. Okay, so these are the pyramids, and clearly you have a left pyramid and a right pyramid. Okay, now, other important structures of the medulla then. Behind the pyramids, you're going to have structures known as the inferior olives, which will be sitting in these sort of spaces here. Okay, so I'll colour these inferior olives here in yellow. Some people will also call these the inferior olivary nuclei. Okay, we'll just call them the inferior olives. Okay, and they are another important landmark of the medulla here. So this is the right inferior olive here, and this is the left inferior olive here. Okay, then behind those, these little extensions off the back here, which I'm now colouring in turquoise, these are basically the connections from the medulla to the cerebellum behind. Okay, and these are known as the inferior cerebellar peduncles. So you have lots of connections from the brainstem to the cerebellum, which is the structure behind the brainstem, okay, which will be sitting here. Okay, and you have the superior cerebellar peduncles, which come off the midbrain and connect to the cerebellum. You have the middle cerebellar peduncles, which come off the pons, which connect to the cerebellum, and now you have the inferior cerebellar peduncles, which come off the medulla and connect to the cerebellum. So these are the inferior cerebellar peduncles here. Okay, right, so now let's put on the chemoreceptor trigger zones onto this picture. They are basically right at the back here. Okay, so you have two of them, one on either side, a left one and a right one. So these are what are called the chemoreceptor trigger zones, or the CTZs for short. So this stands for chemoreceptor, okay, that's the C, and then TZ is for trigger zone. Okay, so chemoreceptor trigger zone. And basically, the neurons in these chemoreceptor trigger zones, they again seem to have 5-HT3 receptors on their surface. And the 5-HT which goes into the blood seems to be able to activate the chemoreceptor trigger zone neurons. Okay, the chemoreceptor trigger zone neurons, it should be noted, have a very leaky blood-brain barrier. Okay, so normally capillaries in the brain have a very, very tight wall, basically, that doesn't allow much to get out of the blood into the brain. Okay, things that want to go into the brain material need to actually pass through the endothelial cells. So basically, the endothelial cells can check everything that moves into the brain. Okay, that's the normal way that it occurs, and that's called the blood-brain barrier. Now, in the chemoreceptor trigger zones, you have a much more less tight blood-brain barrier. Okay, so that many things from the blood can actually go into these areas. So serotonin from the blood can actually enter into these areas of the brain, whereas it wouldn't be able to go into other portions of the brain, because obviously then serotonin is an important neurotransmitter in the brain. You don't want it going from the blood into the brain. That could cause big problems. Okay, but in these special portions of the brain called the chemoreceptor trigger zones, you have a very leaky blood-brain barrier so that they can sample things in the blood, basically. Okay, and the function of these chemoreceptor trigger zones is to filter, well, is to check things in the blood and then decide whether you should vomit, basically. Okay, so they are almost like the equivalent of the enterochromaffin cells, but up in the brain. So the enterochromaffin cells are sampling what's in the intestine, okay, and deciding whether we should vomit. Okay, the chemoreceptor trigger zones are sampling what's coming into the blood. Okay, and of course things that are coming into the blood are generally things from the intestine. Okay, and they are deciding whether we should vomit and empty what's in the stomach, basically, and stop more of these poisons coming in, basically. Okay, so the chemoreceptor trigger zones then, they seem to respond to the 5-HT from the enterochromaffin cells as well, okay, and they again seem to then send messages to these vomiting centers, which are structures very close by the chemoreceptor trigger zones in the medulla, okay, which again triggers vomiting. Okay, so there are these two pathways then by which 5-HT seems to be extremely involved in triggering vomiting, basically, okay, by actually activating vagal afferents directly and thereby stimulating vomiting centers in the brainstem, and also by activating neurons in the chemoreceptor trigger zone, which activate vomiting centers in the brainstem. Okay, so this is the basis of how 5-HT3 receptor antagonists can be used to treat chemotherapy-induced vomiting, because if we block these receptors by giving this drug ondansetron or one of the other ones, okay, we can stop the 5-HT from actually activating these vagal afferents, we can stop the 5-HT from activating the chemoreceptor trigger zones, okay, and therefore we can stop the activation of vomiting. Okay, and that's how 5-HT3 receptor antagonists have a efficacy against vomiting. Okay, right, so we'll call it there for this video, and in the next video what we'll discuss is the Glycine channel in worms, and we'll discuss the major example of a clinical correlate of the Glycine channel, which is the drug Ivermectin, which is used to treat a huge number of different parasitic infections, but one of the most notable that it's used to treat is onchocerciasis, and I'll just talk you through onchocerciasis and the basis of how Ivermectin can be used to treat onchocerciasis.